Okay. Everyone join quickly. Okay. Okay, 1 second. All right. Okay, everyone please confirm me, am I audible to everyone? Just confirm me, am I audible to everyone? Quickly. Okay, no problem, let's start. Okay, now we're gonna start one of the toughest unit in our entire syllabus. Okay, so which is said to be the unit 3. Okay, it is having multiple names. Sometimes we'll use the word, according to our syllabus, fundamental processes. In general, we use the word molecular biology. Okay, sometimes we use the word molecular basis of inheritance. Okay, we will understand how we have given all these names. Why I am saying this is one of the toughest unit is because the way they ask the questions are very tricky, very lengthy, experimental, graphical. Moreover, they will link this particular lesson to the other lessons or other experimental conditions and they will raise the question. And if you want to understand this particular concept, you no need to by heart anything. I can say that 90% of the entire information is conceptual, completely conceptual. Remaining 10% is only you have to memorize the information, you have to remember. Everything is conceptual. Okay, all right, focus everyone. Okay, now tell me, all the students, it's a very much interactive lesson, focus all of you. What is the, if I ask a simple question, tell me Shaila, what is the ultimate function of a DNA? What is that? Very good. The synthesis of a protein, and we generally say that what we can say, it is an information carrier, or we can say that genetic material. The ultimate function of a DNA molecule inside the living cell is synthesize the protein. Why proteins are very important in our body? As I told you multiple times to all of you, in our human body, typical human body I am talking about, we have 37.2 trillion cells are there in our body. Every single cell present in the body, if it wants to perform any kind of a function, what do they require? Protein. Correct na? You taken a best example of a hemoglobin. What the hemoglobin is gonna do? Transport the oxygen continuously. What is hemoglobin? Protein. Can you imagine any any metabolic reactions without enzymes? No. Enzymes are what? Protein. Correct na? Suppose you take an example, if any antigen enter in our body, what the body starts producing? Antibodies. What are antibodies? Proteinaceous in nature. What not? Every single function performed by the living organism require the protein. That protein can be synthesized by what? DNA. That's the ultimate function of the DNA. Now the question is very clear. Then if the DNA having the ability or capability to synthesize the protein, why DNA cannot directly synthesize the protein? Why the DNA is synthesizing RNA as an intermediate? Why it can't synthesize? See, very good. The point is here, always remember, DNA is a highly stable molecule. Right? See, if I am a DNA, if I lost my information and the same information passed it to the next generation, obviously it leads to the mutations. So, in order to protect my integrity, in order to protect my stability, I am going to synthesize an intermediate molecule called as RNA. That the RNA can synthesize the protein later. Suppose example, if this particular RNA got damaged, does it cause any harm? No, because we have DNA. We can synthesize the RNA immediately. Hence, if DNA damaged, we can't synthesize RNA. Even though we synthesize the RNA, it is a mutated. Even though we synthesize the protein, that will become completely non-functional. That's the reason, in order to protect its integrity and stability, the DNA is always releasing what? Always synthesizing the RNA. Through the RNA, it synthesizes the protein. Right. So these three are three common fundamental processes happening inside the cell. What are those? Replication, transcription and translation. Right. So Our ultimate goal of the DNA is what? Synthesize the protein. But it can't synthesize directly. First it gives rise to RNA. Before giving rise to the RNA, it gives rise to DNA. We are all exactly the replica of our mothers or our parents generally. Why? Because the information passes from their generation to our generation via DNA. That's the reason. What do we call that particular process? DNA replication. Copying. A very important statement. From the DNA, what we have to give rise to? RNA. If I want to give the RNA from the DNA, very important statement, it is for the exam. Do we require the entire DNA to give rise to RNA? Then what we require? A small segment, a small piece of a DNA. Now, tell me everyone, why I have taken only this piece? Why I can't take this piece? Because it depends on what kind of a protein I want to synthesize. You're getting the point. So, this part of the DNA, code for this protein what I require. Hence, I will take only that specific segment. A specific segment of the DNA going to give rise to RNA. And that, so synthesis of RNA from the DNA. We call it as what process? Transcription. Can I say DNA mediated RNA synthesis? Correct, na? That is what we can call as transcription. Now mRNA, what it does? mRNA, messenger RNA, it brought the information from DNA. What information it brought? The information to require to synthesize protein. Correct, na? So now that mRNA with the help of amino acids and other, it is going to synthesize the protein. So mRNA mediated protein synthesis, we call it as translation. Can I also say one more word? DNA guided RNA mediated protein synthesis. Correct, na? Without DNA, RNA also cannot synthesize the protein. Correct, na? That's an important statement to be remember for the exam. And before giving rise to RNA, what the DNA is doing? DNA is undergoing the process of replication. These three are very common processes. That's why we can call fundamental processes. Sometimes we use the word called molecular basis of inheritance. Why? The characteristics are passing via molecules. DNA, RNA, protein. That's why we can call molecular basis of inheritance. This is an important point I want you to remember correctly. So now let's understand the location. As I told to all of you multiple times, you have to focus on location. This is only our lesson. Entire lesson is this only. Extreme depth we have to read. It will take 18 to 20 days of time to just cover this. Now listen, our ultimate goal is to synthesize the protein. For that, first DNA is undergoing the process of replication. In our syllabus, we have to read all the three steps: replication, transcription, translation in prokaryotes also, as well as the eukaryotes also. 90% of the time, in order to make the question tough, they may compare the prokaryotes with the eukaryotes. Hence, it's very important you have to read both. Very, very important. Now, very important to know is the location. Tell me, Nihila, where the replication in case of a bacteria it will happen. Tell me, Sheetal, where it will happen, replication in bacteria. Bacteria contain any organelle? No, right? Then obviously it is going to happen in the cytoplasm. Correct or not? In case of transcription in the bacteria, obviously cytoplasm. Correct, na? Translation obviously in the cytoplasm. No doubt. Because, so let's consider this is a bacteria, right? DNA is getting replicated, RNA is getting synthesized, immediately ribosomes will come, synthesize the protein. That's all. Everything is happening in the cytoplasm. But we are highly complex organism. Our genome is extremely complex. Hence, if you consider the eukaryotic organism, we are having a well-developed nucleus. So, in case of our body, where the replication takes place within the nucleus. Correct, na? And transcription takes place also in the nucleus. Translation takes place in the cytoplasm. Okay. Okay. Now you all has to get a simple doubt. Why? See, cytoplasm is the area where the protein synthesis takes place. There is no doubt. Then why whenever the RNA got synthesized in the nucleus, in eukaryotes, it is not modified? In other words, we call it, right, inactive. So in order to make it active, we have to do some processes like capping, polyadenylation and splicing. Then only it enter into the cytoplasm. So what is that process is called as? Post-transcriptional processes, where they are happening within the nucleus. Post-transcriptional processes. That post-transcriptional processes never ever happen in the prokaryotes. Why? What is the reason? Because all of you look into this, I'll show you later. Whenever RNA got synthesized, immediately ribosomes will come and start the synthesis of protein. This is RNA. Immediately ribosomes will come. So transcription and translation are coupled. Both are happening simultaneously. Here, transcription occur in the nucleus. After modification, mRNA enter into the cytoplasm. Then protein synthesis or the translation is happening. So when it comes to the conclusion, in case of bacteria, this is only important: replication, transcription, translation, all are happening in the, no doubt about it. When you look into the eukaryotes, replication, and here is there any post-transcriptional process in prokaryotes? No. In the prokaryotes, post-transcriptional processes are absent. Correct na? Now, eukaryotes, replication, transcription happens in the nucleus. Post-transcriptional processes also happen in the nucleus only. Correct na? Translation happens in the cytoplasm. Correct. Finally, whether it is a prokaryotic or eukaryote, where we synthesize the protein? In the cytoplasm. Correct na? Now, whenever we synthesize the protein, that is also not structurally modified. What do we call it as? Naive protein. Correct na? We call it naive protein, which is inactive protein. So in order to make the protein to become active, what kind of modifications we have to do? Several covalent modifications we will do it. We call it as post-translational modification. The modification which is happening after the synthesis of protein. Now where they are happening? In the cytoplasm, because protein synthesis happen in the cytoplasm. Modification also happen in the cytoplasm. So post-translational modifications are very common in the prokaryotes and eukaryotes. They are happening only in the cytoplasm. Simple. So remember like this: in prokaryotes, replication, transcription, translation, post-translation all happens in the cytoplasm. There is a no post-transcriptional process, means modification of mRNA never happens in the prokaryotes. In eukaryotes, replication, transcription, post-transcriptional, RNA editing also, all happens within the nucleus. Translation and post-translational happens in the cytoplasm. It is super important to remember the what we can say that locations. In the K set, TS set, MH set, 10-15 times the questions were asked, 10-15 times just about the locations. Okay, here itself I will show you half of the lesson. Listen. Now, how exactly we have to read? First we have to focus on replication. In prokaryotes, we will compare with eukaryotes, then we will go to the transcription comparing both, then we will go to the translation, we will compare both. Right. Now tell me, Sheetal, replication means what we discussed? Copying. Copying of the DNA. Now the most important point, what is the major enzyme that is required? What is the enzyme that is involved in the replication of the DNA? Every one of you know that DNA polymerase. Now the question is very clear. Slowly think and understand the point very correctly. DNA polymerase is the one which is involved in the replication of DNA. It is guided. It is mediated by DNA itself. Correct na? That's why we use the word DNA dependent DNA polymerase. Correct na? No confusions at all. Now tell me all of you, what now DNA is going to give rise to? RNA. What is the major enzyme that synthesizes the RNA? RNA polymerase. No doubt. Now RNA polymerase is getting the guidance from the RNA or DNA? DNA, obviously from the DNA. That's why I use the word DNA dependent RNA polymerase. No confusions at all. DNA dependent DNA polymerase involved in the DNA replication, DNA dependent RNA polymerase is involved in the transcription. It is crystal clear, and every student very commonly know one common point, the very important point. All the organisms are having DNA as their genetic material except viruses. Viruses are having what? RNA. So viruses undergo the replication, you know it very well. RNA undergo the replication, that is what the virus will do in our body. At the same time, RNA can also give rise to DNA. Correct na? The process of synthesis of RNA from the DNA, what do we call as? Transcription. Correct na? And the process of giving rise the DNA from the RNA called as reverse transcription. Absolutely true. What is the enzyme that is involved in the synthesis of RNA from the DNA? DNA dependent RNA polymerase. Crystal clear. Now in the reverse transcription, what is happening? Tell me. I am synthesizing what? First tell me. I am synthesizing what? DNA. Correct na? So what is the enzyme that is involved in the synthesis of the DNA? DNA polymerase. Correct na? But that DNA polymerase is getting the information from the DNA or RNA? RNA. Correct na? It is getting the information from where? RNA. So what do we can call the RNA dependent DNA polymerase, also called as reverse transcriptase enzyme. That no one will ask reverse transcriptase. Okay, you have to remember this. RNA dependent DNA polymerase. Correct na? No point of confusion. Every one of you understand the point very correctly. The enzyme which is involved in the synthesis of, very simple to understand. I am synthesizing RNA. So undoubtedly enzyme required is RNA polymerase. Signal comes from, guidance comes from DNA. Hence DNA dependent RNA polymerase. I want to synthesize the DNA from RNA. The enzyme involved in the synthesis of DNA is DNA polymerase, getting information from RNA. Hence RNA dependent DNA polymerase, also called as reverse transcriptase. Correct na? Now where the translation is happening? Cytoplasm. In all the organisms, the location is same. Okay. Is it clear everyone online so far? Please confirm me quickly. Is it clear everyone online? Quickly. Alright. Now what exactly we have to read? We have to start from the DNA replication. DNA replication will take 5 classes. Transcription will take 8 classes. Translation will take 8 to 9 classes. That much big they are. Okay. The more info, even if you are interested in doing PhD also, you can find multiple disciplines in the molecular biology field. It's a very important lesson. Okay. This one and biochemistry. Now let's start with the first one, that is the DNA replication. Alright, continue. So what exactly DNA replication? Copying the DNA, right? You also know, right? Watson and Crick are the one who identified the structure of DNA in the year 1953. What they proposed? Double helical structure. Correct na? Before Watson and Crick, the teacher of the Watson and Crick, that time he was the leading scientist named as Paul. Okay, he proposed triple helical structure of the DNA. Unfortunately, there is no evidence to prove that the DNA is existing in the triple helical. Right, and how Watson and Crick also actually proposed a double helical structure based on what technique? What is the technique? Very good. Based on the X-ray crystallography technique. In the set exam you will get a direct question. Based on what? X-ray crystallography technique. This technique was performed by 2 other scientists. What are those? What is that? Anyone remember the scientists who performed the X-ray crystallography? Every one of you. Online people? Rosalind Wilkins. Okay, James Franklin. Okay, these 2 scientists who performed the X-ray crystallography, but entire recognition taken by the Watson and Crick, but later it became a big controversy. Later Nobel Prize was shared among 4 people. Okay, don't get confused over there. But this is what the double helical structure of the DNA. Anyway, we'll read extreme depth. This lesson I'll explain you very slowly, very elaboratively. Now, how can I define the word DNA replication? Tell me, all of you can observe the picture so clearly. What is happening here? What you observe here? DNA replication is a process, let's see the definition, in which daughter DNAs are synthesized using the parental DNA as the template. So, if I want to synthesize the daughter strand, compulsory what I require? Parental DNA. So, look into this, how the parental DNA, this is one parental strand, this is another parental strand. Correct na? What is happening? The two parent strands are getting separated. We will see that. When both parent strands get separated, what is going to happen? This is acting as a template, this is also acting as a template. This will help in the synthesis of one daughter strand and this will help in the synthesis of another daughter strand. So, both parent strands separated, finally one daughter strand here, another daughter strand here. This is what definition? Chemical definition, sorry, biological definition. DNA replication is a process by which two parent strands are acting as the template, and during the time of replication they are separating and there is a synthesis of the new strand. This is a biological definition. How can I give the same thing in terms of a chemical definition? What exactly the DNA is made up of? If I ask all of you, what is exactly the DNA is made up of? What the DNA made up of? Nucleotides. We generally call DNA is what? Polymer. Correct na? Correct na? Polymer means it is made up of many monomers. What is that monomer? Nucleotide. How many nucleotides are there? Can we count? No one can count exact number. We can give the estimated number only. It was estimated that if you take the DNA and keep on this floor from here planet Earth to planet Mars to planet Earth, it will come 600 times. That much long molecule the DNA was, right? And that DNA is exclusively made up of what? Nucleotides, right? And all the time, every one of you remember, we will discuss elaboratively, DNMP means deoxyribonucleotide monophosphate. N represents number. This is first nucleotide. Every time all of you remember, when one nucleotide adding with the another nucleotide, it always comes in the triphosphate form. What is that? Right. Can you see that? DNTP. We will see that. Deoxyribonucleotide triphosphate. But three phosphates will be joined. Only one will join. How many will be released out? Two will be released out in the form of PPI. Correct na? So we added this also. So it will become N plus 1 and so on. Right, now let's see that. What exactly we all know very well, I'll just quickly give the glimpse. How exactly the nucleotide generally looks like. We already studied, right? The nucleotide is generally made up of how many parts? 3 parts. What are those? Sugar, phosphate groups and base. Correct na? Sugar, what kind of sugar here? Pentose sugar. And pentose sugars are of two types. We all people studied. What are those? Ribose sugar and deoxyribose. What is the difference now? We studied now. What is deoxy means? See, oxyribose means in the second carbon always which group is present? OH group is present. That is called ribose sugar. Based on the sugar only we have given the names to the nucleic acid. If the sugar is ribose, we can call ribonucleic acid, which is RNA. So in the second group, if there is a no oxygen is present, D, no, oxy, oxygen, there is a no oxygen is present, only hydrogen is present, that is what we can call deoxyribose sugar. That is what generally DNA. Correct na? So sugar is what? Deoxyribose sugar. How many phosphates are there? 3. Mono, di, tri or alpha, beta, gamma. What is the DNA is acidic or basic? Why? Because of the presence of the phosphate group. No point of getting confused over there. So then followed by nitrogenous base. What is the speciality of the nitrogenous bases? We all studied. We have 2 nitrogenous bases, purines and pyrimidines. We all know it very well. Purines are always how many rings? bicyclic rings, pyrimidines are the monocyclic rings. Now the most important statement all the students has to remember, I just said everything individually. This is sugar, this is phosphate, these are bases. But if I want to form the nucleotide, all has to come close together. How is that possible actually? That is important. So always there should be a linkage. Correct now? Now all of you understand this is what? Sugar. The first carbon of the sugar can be connected to the nitrogenous base. What is this linkage is called as? N-glycosidic linkage. You read in the 11th standard also. Correct na? Never ever get confused. Always the fifth carbon of the sugar connected to what group? Phosphate group. This cannot be the phosphodiester bond. Always remember that statement. This is called as ester. Right? Widely called as phosphoanhydride bond. Okay? Ester or phosphoanhydride bond. Then what exactly phosphodiester bond is? Phosphodiester bond is the bond which is formed between two nucleotides. Now let's try to understand how the phosphodiester bond will be formed between the two nucleotides actually. All of you please try to understand. I am writing one nucleotide which contain what? Sugar. Sugar connected to nitrogenous base, whether it is a purine or whether it is a pyrimidine. And the fifth carbon it is connected to phosphate groups. Clear? So I am drawing the another nucleotide also here. Contains same sugar, then followed by the nitrogenous base, then followed by the 3 phosphate groups. Now let's understand how does the bonding is actually formed. Now you tell me, this is what sugar? Deoxyribose. Second carbon, what group it contain? H. Third carbon? OH. Correct na? Now we all know very well, we studied multiple times, OH, oxygen, acting as a nucleophile. What? Nucleophile. It is having, can you see that lone pair of electrons? They will do the attack. Right? So oxygen nucleophilically attack the phosphate group present on the other nucleotide. You got it? So 3' hydroxyl group of one nucleotide combine with the or attack on the 5' phosphate of the another nucleotide. You are getting? So hence what happened? 2 phosphate groups will be released out. You are getting? And the bond which is formed here, this is called phosphodiester bond, which we can call backbone of the DNA. Correct na? Now just consider this is only first nucleotide. Consider this is only last nucleotide also. Now just think and answer. This is the first nucleotide. At which carbon phosphate groups are present? Fifth carbon. Now you tell me, does the phosphate groups are attacked by any OH group here? They are free. Correct or not? I call it as 5' end. Correct na? So consider this is the last nucleotide, last. So can I say that the 3' hydroxyl group is obviously free? Correct na? I can call it as 3'. So one strand of the DNA is in 5' to 3', then other strand it is opposite. So which is 3' to 5'. Correct na? That is how the DNAs will be, DNA double strands are anti-parallel to each other. They are not parallel, anti-parallel to each other. They are exactly look like a ladder. Right? If you see from the top view. Fine. Now let's understand, these topics are very difficult now. So now we're gonna start the DNA replication. Now you got a very clear idea what is the definition of the DNA replication, right? So now how we have to read this lesson, how depth we have to read. First, whenever we are studying the DNA replication, all the students understand, first we have to focus on the rules of the DNA replication. Very important one, very good, everyone please continue, that's correct. Later we have to focus on enzymes involved in the DNA replication. Later we have to focus on the exact process. This much complicated it is. First we are only understanding rules, enzymology, process. The question comes from all together. Okay, now what are these rules? Suppose example, if you want to become an assistant professor, you are having certain eligibility criteria. If you are not meeting that eligibility criteria, it's impossible to become. Similarly, if the DNA wants to undergo the replication, it has to strictly follow some rules. If it fails to follow any one of the rule, the DNA replication never happens. Okay, what are those rules? Number one, we will discuss it, semi-conservative. If you get the experimental based question, this is the area where you get the experimental based questions. We'll read it very elaboratively. Then next, it starts at the origin. I'll explain everything. The synthesis always in the 5' to 3', can be uni or bidirectional, semi-discontinuous, requirement of primers, high fidelity. Fidelity is always talking about accuracy. How accurately DNA replication is happening. Now let's discuss one by one rule very elaboratively. The number one, we call it as what rule? Semi-conservative rule. First of all the students has to understand what is this semi-conservative, how I have given this word, from where this word has come from. Listen statements correctly. Whenever the double helical structure of the DNA proposed by the Watson and Crick, they have published their entire research into the world famous journal Nature. You know, they have written one interesting line over there. What they have written, whenever we are studying the structure of the DNA, the structure of the DNA given as a hint. What kind of a hint it has given, we can propose a model by using it undergo the replication. So how they got an idea? By understanding the structure, how the DNA is undergoing the replication. So what is that idea they got? They understood that, listen correctly, during the time of a DNA replication, two parent strands are getting separated. So both parent strands are acting like a template, correct na? So, and to this there is a synthesis of the daughter strands. That is the idea they got, but they have not proved. They understood that while undergoing the DNA replication, mostly DNA follows this. And they have given the name called as semi-conservative, which means one parent and one daughter. You got it? But no one believed in this. Some scientists, they accepted because Watson and Crick are the leading scientists. They only proposed the DNA, so undoubtedly that model is the correct. But of course, many people opposed that model. And they said, why they opposed? What is one of the major reasons? Because there is no structural evidence. There is no structural proof to prove that this model is really working. See this is semi-conservative, where the parent DNA opening, acting as a template strand, there is a synthesis of the daughter strand. But no, some people did not believe that, since there is no evidence. So by opposing this model, other people proposed other models, conservative model and dispersive model. What is exactly the word conservative model? What exactly it is? As the name itself indicating, right, conserving, preserving. They said that entire parental DNA will be conserved or preserved, and it is going to give rise to daughter strand. You're getting the point? Entire parent DNA is getting conserved or preserved and directly giving rise to the daughter DNA. But there's no evidence exactly, we're going to see that. There is another model called as dispersive model. The moment you heard the name of a dispersive model, remember only one thing, cut and patch, which means small parent, small daughter. You can see the color combination, parent, daughter, parent, daughter, parent, daughter, parent, daughter, parent, daughter. That is what model? Dispersive model. One is cut and patch or dispersive, one is the conservative model, one is the semi-conservative model. Now what is the real challenge? The real challenge is that we have to prove semi-conservative model is the only idealized model followed by the DNA for its replication. On the other hand, we have to disapprove dispersive and conservative models were not followed by the DNA during its replication. Right. So to prove this, a classical experiment was happened in the field of biology. Till today, it is said to be the most classical experiment in the field of biology till today. It was proposed by Matthew Messelson and Franklin Stahl. Okay, one of the wonderful experiment it is. In the next class I will show you the animation also. Now let's see how they proved it. Okay, I'll explain it elaboratively. The people who are listening very first time, there is no point of getting confused. You just focus on the concept very correctly, you get a brilliant idea. First, always remember this word. What is our goal? What is our goal first? I have to prove. What I have to prove? Parent strands are getting separated, and I have to prove that. What I have to prove? Daughter strands are getting synthesized. This is I have to prove it. All the time remember my words. Parent strand always heavy. Always what? Heavy. Daughter strand always light. You are getting my point? Parent strand always called heavy strand and daughter strand is called as light strand. But according to the semi-conservative model, what we have to get? We have to get the combination of heavy also, combination of light also, which we generally call it as a hybrid. What is that? Hybrid or intermediate. I hope you are getting my points correctly. So we have to prove this. That means during the DNA replication, only this will happen. Let's see how. They did, very brilliant experiment. All of you just try to understand. First, listen every one of you very carefully. First, they have taken a test tube. The test tube contains a bacterial culture. Okay, it was placed under the N14. What is N14 represents? Which isotope? Light nitrogen, we call it as what? Light, N14. And from here, they have isolated the DNA. Okay, and they start observing the base. When they start observing the base, what they observed only light. But what is our goal? We require intermediate. Intermediate means combination of light and heavy. Correct na? So, but they got only light. Next time what they have done? Again they have taken a test tube, in that bacterial culture is present, that they kept under N15. You getting me? N15. From here also they have isolated the DNA. When they observed the base here also, what they got? Heavy. You are getting me? Heavy means what? Only parent. Light means only daughter. But what we need? Intermediate. Now this time what they have done surprisingly, they have taken a test tube. To this test tube, they have transferred equal concentration of N14, equal concentration of N15. You are getting my point? Plus, they have also added something called as a cesium chloride, which is a salt. Okay. Now the question is that we are having many salt. Why we have added only cesium chloride? One of the major reason is that the density of the DNA, the density of the cesium chloride are almost similar. Density means weight gradient, okay, which is a 1.91 gram per centimeter cube. No need to remember. Just remember that both densities are similar. Now tell me, in this test tube what are there? What are there? Cesium chloride is there, N15 is there, N14 is there. What they have done? They started doing the centrifugation. Centrifugation means rotating at a higher speed for the period of 20 hours. Now what happens? Based on the density, they will settle. If I throw a big stone which is having heavy weight, where obviously it will down. If I throw a leaf, obviously it will float. Same thing will happen here also. Now tell me which is gonna settle down. Obviously cesium chloride settled down, right? After that N15 or N14? N15 will settle at the second. After that N14. Now with the simple thing, they got an idea that I can separate parent strand and daughter strand with the help of density. You got the point? With the help of density, I see, because continuously I am saying which is the parent, parent is heavy and daughter is light. How you confirm? Why I am saying parent is heavy? Based on their density. Now I got a clear cut idea, based on density I can separate. Now with this simple idea, now they started doing experiment on 3 models. Now let's understand which model is very accurate. I will show you the very good animation as well. You will get a brilliant idea over there. Now just very simple point. no point of confusing at all. First understand very correctly. First they have taken N15. Now we are going to check the 3 models. We have taken N15, which is heavy nitrogen, heavy isotope. Now when I take this DNA and when I check the base, what will I get? Will I get intermediate? No. What I will get? Heavy. Wonderful. I will get heavy. No doubt about it. That is absolutely clear. After that, some of the N15 I have transferred to N14. Correct na? N15 and N14 I have transferred. Now from now onwards, I have started taking the sample every 20 minutes. Why only every 20 minutes? Because the generation time of that E. coli bacteria is only 20 minutes. That's the reason I have done it. Now you only observe, no point of getting confused over there. First generation, everyone focus, generation what? 0. Why I am using the word generation 0? Because I have not combined N15 and N14. That's why I call generation 0. N15, what I got? Heavy DNA. Can I say that only heavy DNA I got it? Correct na? Hence 100% of heavy DNA only I got it. Yes or no? Next what I does? Tell me. N15 I have transferred to N14. Now tell me Kavana, what do we get? We combined both. Do we get only light and heavy? Separately what do we get? Intermediate. Correct na? Which we can call hybrid. That is what our model. What is our model? Semi-conservative model. So can I say that in the generation 1 what I got? Intermediate. You are getting my points. Why I have written 100% down? Because it is heavy. So obviously settle where? Down. That is important in the exam. Bottom. Correct na? Why it is intermediate? It is not light nor heavy. Neither heavy nor light. Now second generation. Now you tell me second generation what I got. Can I say that heavy? Where heavy? Second generation. Now what is that? Can you see here when the band is in the middle, what does it indicates? Intermediate. Correct na? This is intermediate. It is a combination of heavy and light. Above, if the band is present above, light. So what I got? In the second generation 50% I got light, 50% I got intermediate. Correct na? Generation 3 what I got? 75% light and 25% intermediate. Fourth generation I got light and intermediate. Can I say every single generation intermediate is repeating? Correct na? Then what you people can say me? Semi-conservative model is following. Correct or not, Shaila? Semi-conservative is following or not? What is semi-conservative? Combination of which is intermediate. It is completely repeating. What is conservative? Entire parent strand will be preserved and giving rise to the daughter strand. Anything you observe? Anywhere. What is dispersive model? Small parent, small daughter, small parent, small daughter. Anywhere you observe? No. Correct na? So which model is idealized now? Semi-conservative model. Okay. I hope everyone clear. Good, very good. Anyway, we'll see little bit animation also, and then you people get little bit very good idea. Many good animations are there, but you follow this very effective animation called semi-conservative. Matthew Messelson and Messel. Oh my God, what is this, man? Animations will really help us in terms of remembering in a very simple words. What is that? Matthew. Just a minute. No, if I later if you see you don't understand at all. What is this, man? There are many videos are there, but I want to show you one very good animation, that's all. Where is that animation? Just a minute. Otherwise, I'll share in the group. You can got it. No, this is for another. Why it is came here? It's okay. It will replicate.
How to get the most of Grammarly free. It's Kayla on behalf of
Which here it will play.
Grammarly to tell you all about the free version of the.
All right, look into this now.
In 1953, James Watson and Francis Crick built their model of the structure of DNA, the principal features of which are still accepted today. DNA consists of two strands that are complementary in base sequences to each other. A cytosine base in one tested to the scientists and one old strand.
Okay, so now you people can observe very clearly the three kind of models. Okay, yeah, you can observe here very clearly these three are the models, right? One is the semi-conservative, second one is the conservative, third one is the dispersive. You can very clearly understand one is parent and light which is called intermediate. Second, what is happening? Only parent is giving rise to daughter. In the third one, what is happening? Cut and patch, a small combination of parent and a daughter. Now I have to prove this correctly, I have to disapprove this. First, what we have to understand? We have to understand the difference between the parent strand and daughter strand. How can we differentiate? Only based on density, correct na? Parent always heavy, daughter is always light.
Semi-conservative replication.
Everyone understand?
No evidence existed to prove that this model was correct. To solve this problem, the key to the Mendelssohn-Stahl experiment was devising a strategy to distinguish between old versus newly synthesized
old strand
DNA.
versus new strand.
They distinguished the two by labeling them with isotopes. They grew Escherichia coli bacteria in the presence of either a heavy isotope of nitrogen, N15, or the ordinary light isotope, N14. After many generations, the DNA in the bacteria contained either the heavy.
So what we got it here? When I take the DNA from the N15 containing bacteria, I am only getting the heavy. Here I am only getting what? Light. But what is our goal? To get the intermediate, right? So now what is the solution for this? We have to combine, correct na? Now see that.
Heavy or the light form of nitrogen, but not both. In this example, the nitrogen atoms in the thymine base are labeled with either the heavy or the light forms of nitrogen. The scientists took samples of each bacterial culture. They processed the bacteria to release the DNA into solution. Equal volumes of the DNA solutions were mixed together, and then this solution was mixed with a concentrated solution of the salt, cesium.
So they only added this.
Built their model of the.
What happened to this?
From your knowledge of example, the old solution of the salt, cesium chloride. The density of the cesium chloride was 1.71 grams per cubic centimeter, the same density as DNA. The tube was placed in an ultracentrifuge
just for balance,
capable of high-speed centrifugation, 140,000 times the force of gravity for 20 hours. At this high speed, cesium ions had a tendency to sediment toward the bottom of the tube, forming a higher density solution at the bottom compared to the top.
And light.
Other substances, such as the DNA, move to the position within the tube that matches their own density. Which band of DNA contains the heavy isotope, N15, of nitrogen? Click on the correct band. With their technique of separating heavy versus light DNA established, the scientists tested the hypothesis of semi-conservative replication. They first grew E. coli for 14 generations in a medium with N15 in the form of ammonium chloride.
Why they have taken only NH4? It is the source of nitrogen.
Growing the bacteria for many generations ensured that all the DNA would be labeled with heavy nitrogen. At this time, they isolated their first bacterial sample. prepared the DNA, and added the cesium chloride for centrifugation. At the same time, some of the bacteria were transferred to N14-like medium and allowed to continue to grow. From this point onward, newly replicated DNA will be made with the light form of nitrogen. They called their first sample Generation 0. After the transfer to light medium, a sample was taken every...
What we got in generation 0? 100% where? What do we get? Heavy. Where the band will come? Below. Banding is very important in the exam, okay? Band always comes below.
20 minutes, which is the generation time for E. coli cells growing at their optimal temperature. The DNA from the samples was prepared for high-speed centrifugation. Meselson and Stahl found the following results. In generation 0,
now you can conclude. 100% is, what is this? Heavy. Generation 1, intermediate. Generation 2, intermediate plus light, intermediates plus light, intermediates plus light. So 2 generations are more than sufficient to prove that semi-conservative model only the idealized model for the DNA replication, right? So this is said to be the one of the most classical experiment that happened generally, okay? It is very important. I'll show you in the next class, the type of questions that people can expect in the exams. Okay, is it clear online people? Just quickly confirm. This is the semi-conservative model. The concept is same, but when they are asking the questions, they'll confuse us and they will ask us. Once you understand the concept correctly, you can answer it so easily. Clear everyone online people? Anyone having any doubt, you can ask me. All right. Okay. Now, what is the next rule? The next rule is, where does DNA replication start? How long the DNA molecule, whether it's a prokaryote or eukaryote, DNA is very long. And it is very important that DNA replication should not happen randomly. It should always happen from a point. The point at which start, what do we call it as? ORC. We call it as origin of replication. Correct, na? No point of getting confused. Now, if you look into the bacteria, E. coli, which is a very small bacteria, the DNA is in which shape? Circular in shape. One origin is sufficient. One origin of replication is sufficient to start the DNA replication. But you take us, we are extremely complex and our DNA is not circular. Our DNA is what? Linear. Hence, one RC is not at all sufficient. What do we call it as? Multiple origins. In the eukaryotes, we don't use the word called as ORC. Okay? We use the word called as replicator. You're getting the point? Instead of using the word ORC, we use the word called as replicator. See, this is one origin, this is also one origin, or this is one replicator, this is another replicator. Like that, more than 150 replicators are there. Okay, since our organism, our genome is extremely complex. Now, whenever we are studying the prokaryotes, every single time we use E. coli as the best and standard example for the prokaryotes. In the exam also, every time they will take E. coli in the classical experiments. Whereas eukaryotes, what we will take in a best example? Yeast. Why only yeast? Why can't we take any other organism? How many chromosomes we have? 23 pairs. Yeast having 16 chromosomes. Some are less equal to the humans. That's the reason we will consider the yeast as the best example. In case of eukaryotes, what do we use the word? Replicator. Especially in the yeast, we are having other name for this replicator. I hope you are getting me correctly. We called ARS. I'll tell you in the processes. Autonomously replicating sequences. What is the meaning of autonomously? Are they dependent on? No. So can I say that all are activating simultaneously? That is also a question. That's a very important statement to be noted down. They are all autonomously replicating sequences. So in case of a prokaryotic organism, correct. In case of a prokaryotic organism, there is only one ORC. In eukaryotic organism, there are multiple ORCs. Now, this is said to be one of the most important rule which makes us a lot of confusion, and if the question comes from here, 90% you will get a lot of confusion. So in order to avoid the confusion, first you require a better understanding of the concept. Direction of new strand. According to the rules, every DNA strand which is synthesizing newly, that is daughter DNA, all the time synthesizes only in which direction? 5 prime to 3 prime. It is fixed. Now we have to prove why only 5 prime to 3 prime, why not in any other direction. That we have to prove. Before that, you all people has to understand one crystal clear point. What is the major enzyme involved in the DNA replication? DNA polymerase. Remember my statements correctly. DNA polymerase enzyme read the parent strand all the time. I am talking only parent strand. In which direction? 3 prime to 5 prime. You are getting the point. Parent strand in 3 prime to 5 prime, obviously daughter strand has to be synthesized in the 5 prime to 3 prime only. This is only one reason, okay. The other reason is actually the presence of 3 prime hydroxyl group. This is the major reason. Now we are going to justify it. Let's try to understand. Very important concept it is. Now tell me, Kavana, here. This is what? 5 prime to 3 prime, correct na? At the 5 prime and how many phosphates are there generally? 3. Very good. Alpha, beta, gamma. What is this group? 3 prime contain what? OH group. Wonderful. Now tell me, what happened now? New nucleotide is coming. New nucleotide comes with how many phosphates? I always told to you, new nucleotide comes in the form of DNTP. How many phosphates? 3 phosphates. Crystal clear. Now what is happening? Tell me. This OH group will having oxygen as their nucleophile will come and cut what? One of the phosphate group. Correct or not? How many gonna released? 2. In the form of PPA. No problem. Hence it got settled. Correct na? No problem at all. So nucleotide added successfully during the proofreading. What is proofreading? Checking the errors. While checking the errors, DNA polymerase identified that whatever the nucleotide it added, that is a wrong nucleotide. Now what should we do now? 100% we have to remove it. Correct na? I have removed it. I have to replace with healthy nucleotide. If I want to add this healthy nucleotide, can I say that compulsory again I require 3 prime hydroxyl group? Correct or not? So anyway 3 prime hydroxyl group is available. What I have removed is nucleotide, which is the wrong nucleotide. I have not removed 3 prime hydroxyl group. Hence healthy nucleotide will be added. You are getting the point. This is what happens generally. Now let's understand the hypothetical situation. It never happens, but to discriminate it I am just telling you. You imagine every single time whether it's a DNA or RNA, nucleotide always added at which position? 3 prime hydroxyl position. Now I want to prove to all of you why nucleotide cannot add at the 5 prime group. That is what I want to prove. Now this is the new nucleotide having how many phosphates? 3. At the 3 prime group what it is having? OH group. Wonderful. Now don't get confused. This is hypothetical. Okay. Now this OH group, you know what is happening? It will go and cut this phosphate. I hope you are getting me correctly. Correct na? Then how many phosphates are released out? 2 phosphates successfully released out. Clear? Huh? Now there is no confusion. You can see, right? Nucleotide added. Correct or not? Now again proofreading is happening. What happens during the proofreading? I have removed this compulsory. Right? It came out. Correct na? I have to add which nucleotide? New nucleotide. If I want to add the new nucleotide, here what group is present? OH group. What the OH group has to do? It has to cut the phosphate. Is there any phosphate is present here? 3 phosphates. Correct na? You are getting my point correctly. Hence, you can't add the nucleotide here. Once again look at this Kavana correctly. Whomever did not understand, follow this correctly. This is the 5 prime to 3 prime. We are proving why can't we add the nucleotide towards the 5 prime direction. This is the first nucleotide having the OH group. This OH group nucleophilically go and cut the one of the phosphate group here. Then these 2 are released in the form of PPA. Then this entire nucleotide will go and attached. Crystal clear. During the time of a proofreading, what I have done? I have removed it. correct na? Now I am going to add this. This is having what group? OH group. If this oxygen wants to show the nucleophilic activity on the 5 prime carbon, how many phosphates should be there? 3. Is there any 3 phosphates are there now? Because we removed it out. Hence, this energy, this reaction cannot be proceed. You are getting? Hence, it was proved that the new strand synthesis always happens in the 5 prime to 3 prime direction only. Okay, it never ever happen, even hypothetically also, in the 3 prime to 5 prime direction. Got it? Everyone online? Is it clear online, people? All right. Okay. Only for 10 days, I'll do it in YouTube. Okay. So that's all for now. Tomorrow I'm just telling you, 10:30 to 9:30 to 10:30 we'll continue the plant physiology in the YouTube only. Okay. Then 10:45 we will start, and 11:45 we will do it in the YouTube only, Mol Bio, and we have 4 classes left over for the immunology. Okay. 12 to 1 we will do it in the app. Okay. Immunology. Okay. So if I finish this, I'll continue the genetics. Okay, only these 2 lessons just for 10 days we will do it in the YouTube. Okay, clear everyone? All right, we'll stop now. Okay, we'll continue in the next class. Okay, so keep revising it. First let me complete 2 to 3 classes, then automatically you will get an idea, and keep reading and keep understanding. Okay, whenever I am solving the questions, trust my words, it is not at all what you read. It is all about how you apply. Okay, so you require a brilliant understanding of the concept rather than remembering it. You have to think reality, you have to think in a practical approach. Then only you can answer the questions from this one. Out of 6 part C questions are coming in the exam, trust me. See, there are 2 kinds of situations are possible here. Out of this 6 questions in the part C, trust my words, 2 questions are so easy. If you identify the 2 questions, you are happy. If you are unable to identify, you have to try to solve that 4 questions, which are very tricky. Okay, your entire effort finding these 2 questions and making them correct. Okay, all right. I can say that one of the deciding chapter in the paper. Okay, so we have to read extreme depth. I hope it's clear for everyone. Correct, correct. Okay, that's all for now. I'll see you in the next class. Okay, thank you. Yo, 12:30 I have another. Okay.